meet of our seminar series now. It's my real pleasure to present my friend David Barbie. Dave is an associate professor at Harvard Medical School and Dana-Farber. In truly inspiring fashion, he overcame handicaps of both an undergraduate and medical education at Harvard to become a real expert in lung cancer biology. He worked as a postdoctoral fellow in Bill Hahn's lab and has had his own lab for several years now. Most pertinent to what I think he's done a lot of things, but I think most pertinent to what he'll be discussing today is looking at something that is, of course, really hot, and that is developing ways to better understand who will and who will not respond to immunotherapy. And he uses a very functional approach, which means it's very appropriate that he's talking to us today. So take it away, Dave.
Thanks so much. Can you hear me and see my slides okay? Oh, can you hear me? Oh, hold on a second. Yes?
Yeah, we can hear you. Yeah.
Okay, great. Sorry, I hid the thumbnail. So yeah, thank you very much, Tony, for that kind introduction. And yeah, it's a real pleasure to speak here. And I'm going to talk about work that we just recently published looking at RNA sequencing analysis of ex vivo immunotherapy response. These are my disclosures. And so I don't think I have to introduce to this group too much the contrast between genomic precision medicine and functional precision medicine. But as many of you are all familiar with, the classic example, at least in lung cancer, which is my field of genomic precision medicine, is identifying an EGFR mutation and treating cancers with an EGFR tyrosine kinase inhibitor. And while this has been incredibly successful, has revolutionized the field, this has not generalized even within lung cancer, other types such as squamous cell or small cell. And as I'll show you in a moment, even though we understand this, there are subtleties that require more detailed approaches. But I think immunotherapy is even more complex. And what this really relates to is the dynamic nature of the immune system, which I really think is underappreciated since many groups still focus on static snapshots, going to the levels of even single cell RNA-seq immune atlases that are all basically conducted at one point in time. And so the point I like to make is that even PD-L1 itself is a dynamic biomarker. It's induced by interferons in the tumor microenvironment. And then the actual therapy itself, which is intercepting PD-1 from binding to PD-L1, results in a dynamic process of T-cell infiltration. And this occurs over time. There are multiple other cells in the tumor microenvironment that are impacted by this. There's local lymph node trafficking, antigen presentation. There's dynamic upregulation of compensatory immune checkpoints. And as I'm going to talk about, differential responsive tumor cell subpopulations. So these dynamics are not captured by a single point in time. And coming back to the EGFR example, we still don't understand why do some patients have dramatic responses, even with the best therapies now, osimertinib. About 30% of patients do actually have durable responses after 3 years. But why do those 30% have durable responses, whereas 70% don't? And one answer is that there's also adaptation over time, something called drug-tolerant persister cells, which again, I think many of you are familiar with, but I'll also highlight further throughout the talk. So another point that I like to focus on is that clearly many groups are focused on developing functional models using patient-derived xenografts and organoids. And while these are fantastic models for targeted therapies, when you actually want to study the immune microenvironment, what's still done currently are single point in time experiments. So for example, for single cell RNA sequencing or flow cytometry, what's happened is the explanted tumor is digested, single cell suspensions are created, there's cell profiling, and then we get the single cell RNA-seq data. But current methodologies to develop living tissue, what happens is, you either put an immunocompromised mouse or start to grow an organoid, you're basically enriching for tumor cell populations and losing immune populations. And in the PDX model now, you have mouse-specific immunity. And while a number of groups are making humanized mouse models, still there's many caveats with that interaction. So what we've developed is an ex vivo approach to do a short-term assay, where basically you take either the fresh tumor from either a patient or a mouse, and we call these either patient-derived organotypic tumor spheroids, or if the murine model is a syngeneic model, which I'll show you the bulk of this talk, we call it MDOTs, or for targeted therapies, we create from PDXs called X dots. But basically what we do is we take the fresh tumor, we digest it, as I just showed you on the last slide, as is done for baseline immune profiling, but we only partially digest it, so that we do get a fraction of single cells by using a filter. But above a 40 micron filter, you capture larger pieces of the tissue. And I will say now we're actually using every piece, even these larger, greater than 100 micron pieces. But what we focused on to date are these spheroids that you capture between the 40 and 100 micron filters. And what we've shown extensively in prior work is that these contain multicellular components, not only tumor cells, but stromal cells, immune cells. And I'll get into greater detail. And so what we'll often do is we'll flow profile these as the input, and then we load them into a microfluidic device developed by Roger Kam that has a collagen matrix in the central chamber. And basically this just shows you that these spheroids over time grow and expand. And the advantage of this model is then you can do multiple different readouts. You can image. And what we had shown previously is live dead with a perturbation and cytokine analysis of conditioned media. But what I want to focus on today is developing higher order readouts using RNA sequencing. And so this is just, again, the background of our publication from a couple years ago now. To validate this, we focused on syngeneic models. And while we've done a lot of work in patient specimens, as I'll show you in this talk as well, the advantage of the syngeneic system is that you know responsiveness to checkpoint blockade, and then you can actually validate your results back in the animal. And so MC38, which many of you may be familiar with, is a syngeneic colorectal cancer model that has microsatellite instability and is thus a known responsive model, not a cured model, but a responsive model to PD-1 blockade in vivo. So in our prior work, we use this as an example. If you generate these tumors after about 2 weeks, harvest them, create these spheroids, and then culture them for 6 days in the presence of either an IgG isotype control or an anti-PD-1 antibody, use live dead imaging with dead in red here, you can see that we see killing. And this is just quantification of this result using multiple of these devices and averaging over the entire well, that you can see there's a dose-dependent anti-PD-1 and time-dependent anti-PD-1 killing relative to an IgG control. We did a number of other controls. For example, if you just take the cell line and make spheroids and put them in these chambers, you don't, as you might expect, you don't see any anti-PD-1 effect. But we also showed that if you use the M dots and treat with a CD8 neutralizing antibody, you can reverse this effect. So what we wanted to explore is taking this very well-defined system, this MC38 model. Can we do RNA sequencing? And basically what we did was just what I showed you on the last slide. We generated tumors from the MC38 model. We created these M dots, loaded them in the central chamber, and then we treated either with our IgG control, we treated with interferon gamma as a positive control. Since we were looking at transcriptomics, we wanted another parallel control, and then anti-PD-1. As well as the CD8 neutralization experiment, which I described. And you can see here that we saw the same degree of killing as before with anti-PD-1 that was reversed with anti-CD8. Interferon gamma, this cell line was not sensitive. MC38 spheroids were not sensitive to interferon gamma in terms of cell death. So what we did was we basically did the exact same treatment I described, but in parallel wells at day 6, we simply add, you can just simply add the lysis buffer for RNA. Even though there's collagen there, you basically just add the lysis buffer, and then you can just simply perform bulk RNA sequencing. And so on the left, I want to draw your attention to what happened when we compare the interferon gamma condition to the bulk, so the IgG bulk RNA sequencing data. And we ran a volcano plot. And what we were very gratified to see then in this volcano plot is that the most highly significantly upregulated genes are all known interferon gamma targets. We see CXCL10, which is a classic downstream chemokine, and all of these GBPs are known interferon gamma target genes. And so this gave us confidence. And when we ran gene set enrichment analysis, the #1 GSEA gene set is interferon gamma response. So we were very curious now, well, what happens when you compare the anti-PD-1 treated samples? And we saw a very different set of genes and signatures. And what these related to were inflammatory pathways, but actually the #1 pathway was a TNF alpha NF kappa B signature, and the number 2 was an EMT signature. And I'll point out that even though IL-6 signatures weren't captured by this, we saw multiple components of the IL-6 signaling pathway. This is just, we were limited here by what's in the hallmark gene sets. But you can see LIF, which is a known IL-6 related cytokine, as well as SOX3, which is upregulated, is a negative feedback mechanism in response to IL-6 pathway activation. So we're very interested in this. And this harkens back to what I was mentioning about the concept of EGFR drug tolerant persisters, because it turns out that when you kill the majority of EGFR sensitive cell lines, you get subpopulations that are quiescent, they undergo EMT, and actually IL-6 signaling has been implicated in the survival phenotype of those drug tolerant persister cells. And so to start to look at this further, because I mentioned that actually what we're profiling here are the residual cells, is we decided to see can you start developing even higher resolution methodology in this system, and that is single cell RNA sequencing. Now, the challenge with single cell RNA sequencing with our technology is that these spheroids are embedded in collagen. So whereas bulk RNA sequencing, you can just add the lysis buffer, here you actually have to digest at the end of 6 days, culture with collagenase to liberate the spheroids, and then to get them to the single cells, because the spheroids still cluster together, we have to add a trypsin step. But we are successfully able to generate single cells for profiling. But what I'm not showing you is that the immune cells have difficulty surviving this. We could show that whereas we have immune cells at the end of treatment, that this process, we deplete immune cells, which actually was sort of beneficial for these analyses, that we could be confident we were focusing on tumor cells. But it's certainly something that we are optimizing currently. But here, and I will say there were some stromal cells that survived here too, which is important, but a very small fraction. What was gratifying here, though, is that we could be confident that most of these cells were the MC38 residual tumor cells. And when we ran the high dimensional reduction analysis, this is through the TISNE analysis that many of you may be familiar with for single cell RNA-seq analysis, we saw significant clustering. And if you just look at the two conditions, the IgG versus anti-PD-1, we saw a very clear separation. And so what we're very interested in is what defines these 4 different subdivisions of the anti-PD-1 treated cluster. And what we found is that actually what was dividing this cluster 1 and 2, the dominant clusters, were actually cell cycle genes, the expression of E2F target genes. And cluster 2 had low E2F target genes, low mitotic related genes, suggestive of these cells being stuck in a quiescent cell state. And when we projected that EMT signature that we derived from the bulk RNA-seq analysis, this is where we saw this signature enriched, was in this cluster 2. And so this got us to start to think, is it that what's happening is that when you actually profile this response over time, you're uncovering a subpopulation of MC38 cells that can adopt this very similar what we called immunotherapy persister population, IPCs, that are resisting CD8 T cell mediated killing. So to study this further, we needed markers of this cell state. And so we did another analysis of this data where we took this signature, which marked this cluster 2, and we did a supervised analysis to say, okay, if we take this signature and we look at the single cell data, and we say which cells have the highest expression of this signature versus the lowest expression of the signature, can we identify marker genes? Now, of course, some of these genes here were actually in the signature itself. So of course, this was tuned by the signature input. But what jumped out of this were genes that weren't in the signature. And there were 2 genes, SNAIL1, which you may have seen on the original bulk RNA-seq, we were very interested in because it's clearly implicated in EMT, but not just EMT, but the stem-like partial EMT state that Bob Weinberg has talked quite a bit about, but as well as this marker SCA1. And SCA1 has actually been well described in the mouse stem cell literature, mainly on hematopoietic stem cells. And actually Carla Kim, who trained in Tyler Jack's lab, has shown that this is actually can mark lung cancer progenitor stem cells. And so this has not actually been described in these syngeneic models, but we wondered were the tumor cells actually marked by this subpopulation. So the advantage of SNAIL-1 is that there are good reagents to test actually in vivo. Was this just an artifact of our ex vivo system? Could we detect these SNAIL-1 positive cells in vivo post PD-1 treatment? And indeed, this is through in situ hybridization. It's particularly in the areas of cell death, the perinecrotic areas, we saw the emergence of these SNAIL-1 positive cells. And so again, in the interest of time, I can't show you all of the data. But the advantage of having SCA1, one possibility, as I mentioned, could this just be in stromal cell populations, is we actually went back to the cell lines themselves, the MC38 cells, and we also did the same treatment in another model that's actually more resistant to checkpoint blockade, CT26. And if you look at CD44, which is one known mesenchymal marker of these types of cells, these colon lines are all positive for CD44. But what's very interesting is that consistently we could detect a very small population of these SCA1 positive cells pre-existing in this MC38 cell line, and that was consistently higher in CT26. We could also use this to now actually, by sorting the SCA1 positive cells, we did whole exome sequencing to show that these were actually still MC38 cells, not some sort of contaminant. But what we did was we took the conditioned media of these cells. We noticed that CT26, which had higher levels and when we purified them were persistent over time, we noticed that there were many cytokines that were higher in CT26 relative to MC38, and one of them was IL-6. So we actually tested a large panel of these cytokines, including LIF, because we thought that was one that might be involved in this phenotype. But actually the only one that actually scored in actually now expanding these SCA1 cells within MC38 and in many different syngeneic models was IL-6. That what we're showing here is that if you purify these SCA1 cells, try to culture them over 4 days, they go back down to this baseline. But if you now culture them in the presence of IL-6, you now consistently maintain a higher level of these SCA1 positive cells. In CT26, where we purify them, they fall back to a higher baseline. If you now do this in the presence of IL-6, it's now 78% of the population. And we saw this even in lung models that had high levels of these SCA1 positive cells. This worked across cell lines. So clearly, IL-6 is playing some role in expanding this cell population. But I also mentioned that TNF-alpha was the top gene signature that scored. And so we wondered what was the role of TNF signaling? And to our somewhat surprised, but maybe in retrospect not surprised, when we profiled these SCA1 positive cells, we found that while there were NF-kappa B survival genes that we detected in our book that were activated, namely BERK2 and BERK3, that there were many death genes that were also activated by TNF. And in fact, in contrast to IL-6, in this experiment I showed you that could expand these cells, we saw sensitivity of these cells to TNF. And in fact, there's this sort of yin-yang response that some cell lines like MC38 that were very sensitive to TNF, you couldn't overcome the TNF death signal with combination with IL-6. Whereas in CT26, which had a more robust percentage of these cells, IL-6 was overcome the concomitant treatment with TNF. But what this led us to think is, could there be a vulnerability here? That if these cells are able to persist, and the other thing I'll mention is that these genes actually have now come out in other studies. Nick Haning's group, who looked at markers of in vivo CRISPR screens of sensitivity to PD-1 blockade, these genes came out also as if you knock them out, sensitized to PD-1 blockade. And then other work has shown that that there's a TNF vulnerability, both in tumor cells and now also other work showing that if you antagonize these genes, you can activate immune cells. So this is also falls within a larger literature. But this gave us the hypothesis, well, what if you now treat with these antagonists? So one of the antagonists that we focused on is one that was been developed by Novartis, LCL161. It's a BERK2/3 antagonist. And so here's the data that I described to you, just the raw flow counts, that if you've purified this fraction and let it go for 96 hours in MC38, it goes back down to this percentage. You can boost that with IL-6, TNF drops it, and IL-6 is not dominant over TNF. But now if you do this all in the background of LCL-161, you basically completely wipe out cells, period, when you co-treat with TNF. And now actually you're able to completely eradicate even the present, you blunt the effect with IL-6, and now you further sensitize this. And in our CT26 model that was more resistant, now you actually further sensitize these cells, again, with the addition of LCL-161. So this led us to the treatment study that while, as I mentioned, MC38 responds to PD-1 blockade, and this is just, again, showing these individual tumor plots, you very rarely can cure these animals. So what happens now if you treat, sorry, this is the LCL alone. This is the anti-PD-1 alone, that you see a very similar pattern that you shift the curve, but you barely cure the animals. Same thing happens with LCL treatment alone. However, the combination synergizes in MC38 to lead 7 out of 11 complete responses, thus suggesting that changing this TNF cytotoxicity threshold can help eradicate these immunotherapy persister cells and lead to a more durable ICV response. So in summary, what we're excited about more generally is the ability to develop this methodology further and to perform dynamic assessment to a variety of different immunotherapy treatments over time. And instead of just cytokine profiling, which we focused on to date, but actually do bulk RNA sequencing, which is straightforward to do, but also single cell RNA sequencing to really look at what's happening in different immune populations over time with these immune perturbations. But I think the biology that we're interested in here is that this dynamic analysis identified that very similar to drug-tolerant persister cells, even when you have a successful immunotherapy, there's immunotherapy tolerant subclones that can mediate resistance. And as I just showed you, we think that BRK2/3 antagonism is one way to co-opt the TNF sensitivity of these cells to promote apoptosis. And one sort of parallel that I think is good to make is that in the EGFR drug-tolerant persister cells, and I know Tony's done some work on this as well, also in collaboration with us, that there are apoptotic vulnerabilities that are more related to the BCL2, BCLXL pathway. Whereas what we're seeing here in the immune contexture is an apoptotic vulnerability related to an immune cytokine, TNF. And so in addition to focusing on this, targeting this, that simply combining BRK2 and 3 antagonism with checkpoint blockade may not be enough, and that if one could also drive, use IL-6 as a biomarker and focus on tumors that are lower in IL-6, but also focus on driving TNF in the tumor microenvironment, that we think this would also enhance this combination and generate more broader responses. So that's what I have. This was a large collaborative work between my lab and the Belfer Center at Dana-Farber. Cloud Palitz, who's the head of the Belfer, has worked closely with me, particularly on this model system. Kartik Sehgal was a very talented postdoctoral fellow in my lab who led this project together with members of Cloud's team. And I just wanted to highlight some our bioinformatics collaborators in my funding as well. So thank you very much.
Great, that was beautiful, David. And I think it's another great, as we know, with so many complex systems that you get so much more information if you actually can watch the system evolve following a perturbation. This is a great example of that. I'll ask a question and then refer to the chats. So maybe you can make, so this LCL-161, you call it a BRC, a BR domain inhibitor. It also goes by names like SMAC mimetic and IAP inhibitor. Maybe you could just detail a little more explicitly the connection between this class of drugs and TNF alpha signaling.
Yes, and like I said, we are not the only ones to focus on this. In fact, there's been other sort of, there was a Nature screen that just came out, paper and Cell as well, looking at sort of the paper and Cell at the NKI was looking at interferon gamma independent mechanisms of immune killing. And this scored as well, actually, along with one of my favorite genes, TBK1, which I've studied for many years. And I think people are appreciating that there's a number of these pro-survival, that TNF has always been known to have, that's why it's called tumor necrosis factor, these dual pro-death and pro-survival signals. And so I think there's an increasing recognition that if you can identify which ones to remove, that you can shift towards the death impact. So there's other ones, TBK1, IE is one of them, and others that are candidates for this.
But if I'm not, just to make it a little more explicit, it's probably has something to do with the role that CIAP1 and 2 can have in regulating TNF alpha signaling downstream of TNF alpha death receptor, right?
Yes. And you know, the problem is, you know, I'm not the level of expert that you are in apoptosis, but yes, no, and there's probably even further experts as well on actual TNF signaling and as well.
Yeah. I mean, I don't want to belabor it, but just because I think that this is an underutilized, this is a drug family that has a lot of activity, a lot of biological activity, but no one's really figured out how to use them to actually induce good responses in people with cancer. And I don't think we should ignore this class. I think this is very exciting what you're showing here, but I think there's other contexts where they're potentially important drugs.
Yep.
There's a lot of questions. I'm not sure we'll get to them all. Perhaps you can make independent contact with David if we don't get to all of your questions. But one question, what are your thoughts about the 4 out of 11 cancers that did not respond to the dual inhibitory approach?
Yes. I mean, I think in general, with you see so much stochasticity in these syngeneic models. I mean, ultimately, what you really require beyond what I just described is a clonal T cell, clonal CD8 T cell that recognizes tumor antigens effectively, you know, multiple of those, and that's a stochastic process. So what I think is happening is that in the ones that did respond, in addition to eliminating these cells, you really, through cross-presentation and dendritic cells, had really robust effector T cells that were able to then clear everything out, especially when you eliminate these persister cells. But in the other ones, that in a stochastic fashion, we may not have found the right CD8 T cell effectors. Okay.
There's another question. The different models you studied seem to have distinct baseline percentages of SCA1 positive cells. So is this difference to their cytokine profile, or to what do you attribute this difference?
Yes, well, in part, the adding the additional cell lines was done in response to reviewer. So we actually didn't go back. So we discovered we performed the Luminex on the MC38 versus CT26, and we saw the difference in IL-6. We then, in the additional syngeneic models, we didn't run Luminex on all of those syngeneic models to actually correlate the baseline to answer that question. So I can't answer that question, but they were all sensitive to IL-6 when we added it externally.
Okay, great. One last question. I'm afraid the other ones maybe I'll have to contact you differently. Many of the T cells in the tumor that may be responsive to PD-1 blockade will be non-tumor reactive, but responsive to other things like viruses, self-antigens, and so forth. And can you distinguish these from your single cell RNA-seq?
That is another excellent point that I'm appreciating more and more through my collaboration with Ellis Reinhertz, that this is probably one of the major issues, that you're recruiting irrelevant T cells. And the answer is, we've done this not in the device, but in vivo using TCR sequencing. And yes, you can resolve clonal TCRs, but the other answer is no, that you can't go, well, even though some people claim, you can't go from an alpha beta TCR to a target. That's a major challenge. And so many other people are trying to develop methods, improved methods to do that. But yeah, you can look at T cell clonality, but how to know what these are reacting against requires a lot more.
Right, a lot of work. I think, thanks a lot. I think we'll leave it there. Arko Dasgupta had a question. Maybe you could answer it in the chat if that's available to you, David, or otherwise. It's my pleasure to introduce Dr. Drs Lohse for our second speaker. She has a PhD from the University of Zurich and did her postdoctoral work at Princess Margaret Hospital in Toronto. where she focused on characterizing PDX models and testing novel pancreatic cancer therapeutics. She's continued her postdoc work at Michigan and is now an assistant scientist at University of Miami. And she's really focused on using ex vivo drug sensitivity screens to try to provide personalized treatment plans for platinum-resistant ovarian cancer. So please take it away, Dr. Lohse.
Thank you. I first have to warn you, so my 4-year-old thinks that the names in my talk are really boring and that the names in David's talk were way cooler, but I hope I'm not going to bore you too much. She was very specific about that. That's the thing with 4-year-olds that do home learning. So yes, I'm going to talk about drug sensitivity testing for the treatment stratification of cancer patients. And I'm going to talk mostly about the clinical implementation in liquid and solid tumors. We have learned a lot over the last 4 years in doing that, mostly because doing it from a scientific standpoint is very, very different than suddenly dealing with the clinical entity behind it and dealing with live patient samples that need to be collected, shipped, preserved, tested, and then following all the clinical protocols actually being reported back on. I'm not sure why it's not. No, I have no conflict of interest to report. So this is a quick one. So let me tell you about our drug sensitivity testing platform to start off before I go into the actual clinical details. So the platform is developed for clinical use. And again, this is very, very different. It requires a lot more steps than you would usually take if you just use it for a research purpose on mouse or even archive human samples. So everything we do starts with patient recruitment in the clinic and all the steps you need to take there, such as getting consent, making sure that everybody knows that we are expecting a sample and that they need to take it, that they are prepared to take it and we are prepared to receive and test it. This is all done before the surgery date is even planned or the acquisition date is planned. We then have a sample taken in the clinic, and generally that is a standard of care procedure as part of our clinical trial. The sample is then, after it's taken in the clinic, shipped to us, and that can either be local or within the US. It arrives in our lab, hopefully everything is well and it's alive. And then we have to purify the cancer cells out of that sample. And since we are getting a variety of different samples, surgical, clinical, all of that, it makes it very challenging to have all of the protocols for every single cancer type established. We then do the drug sensitivity testing, which is actually the easiest part of the entire screen because we know how to do that very well. We have done it for a long time. Where, sorry, it is not allowing me to change my, no. Where we are seeding the cancer cells on 384 well plates, a whole bunch of them. And we are exposing them to our standard library or to patient-specific libraries if there isn't enough tissue to run the standard library. Currently, our standard FDA-approved library is 250 compounds in size, and all of the compounds are available on compassionate care. That is very important because we want to stratify patients, and not having an easy access to these drugs in the clinic is really hampering the efforts. So currently we are only using drugs that are available to the patient on compassionate care or on specific libraries where we have cleared the clinical availability of that specific compound prior to running the screen. It doesn't make much sense to run the screens on the patient samples if we can't actually get the compounds into the patients. All of the compounds are on the library in dose response, and we're doing an 8-point dose response right now. So we are getting the dose information on all of the compounds as well, which is very important for the analysis. And then we have quality control. Our quality control regimen is fairly extensive because These are patients, they're getting potentially a harmful drug. So we have to make sure that the screen is running well every single time, and that it's consistent within the screen between different plates. And that is achieved through positive and negative controls, duplicate compounds, and all kinds of checks and balances that make sure that the plates are consistent and the screen has run well. We then come to the analysis, and that is, the analysis was developed by Dr. Hassan Al-Ali, who is actually in the audience as well. And he is at the forefront of making the algorithm, extending the algorithm, and doing the majority of the analysis for the patients. And what you can see here is some of the readout of the analysis. So what we are most interested in is the DSS score. And you can see that different compounds have different DSS scores. And the DSS score is a single numerical value that allows us to rank order compounds by efficacy. This includes information on the IC50, on the area under the curve, and also on the total cell killing, because ideally we want to find a compound that works very well over the whole dose range and achieves 100% cell killing, which is not the case for most compounds. So integrating all of this information into a single numerical value allows us to develop drug sensitivity profiles that are specific for this patient at this point in time. So of course, if you're testing a sample a month down the road, it is going to look different than it does at this time. Here you're seeing the rank order of the compounds from highest, which we believe has the highest efficacy in cell killing and on the screen, down to the lowest. We have a threshold, so only compounds that are above the threshold are reported in the clinic, because everything under we don't believe has enough chance of working clinically. We then also include a normal tissue control. So the results of the specific patients get normalized to that normal tissue control to give us an idea of how normal tissue toxicity for that compound looks. Including the normal controls gives us the chance to propose a compound for treatment that is specific for the cancer and shows little tumor toxicity, little normal tissue toxicity. So a very high specificity for the tumor. This is specifically important when you're dealing with patients that had prior toxicities or have been on continuous treatments, are not in good health. The next step then is again quality control. As I mentioned, quality control is a big thing for us and really drives every single step in the DST screen. Once the screen in its entity has passed quality control, we are transmitting the results to the PTC, the Personalized Treatment Committee, which consists out of doctors that are here in Miami and are considered experts in their field. So we don't make any treatment decisions based on the screen. The PTC does that, and they're integrating the patient information as well as the DST screen to then come to a treatment decision and start the assay-directed therapy. So while the DST screen is a big part in making the stratification decision, it is not the only part, because we all know patients are complex and a lot of information has to be integrated in order to ensure that the patient receives the correct treatment. We have learned that turnover time is extremely important because the patients can wait months to actually start the treatment. So we are currently committing to a 14-day turnover time. That starts the minute the sample gets into our hands, it arrives in the lab, and it stops the day the treatment is actually implemented. So it is a very short time frame. But right now, the patients that we are looking at are relapsed refractory and they're fairly sick. So we have to do this in the shortest amount of time possible. We first started it in liquid tumors because obviously it's fairly low-hanging fruits for this kind of analysis, because biopsies are routinely available. The patients are getting them every 3 months in any case. So you can continuously get samples if you need them. All of the patient samples, especially in the relapsed refractory space, have really high disease counts, so high blast counts in the bone marrow and in the peripheral blood. So we don't need a huge amount of tissue. And the majority of these patients don't have any treatment options available. So they're very open to trying something that is potentially a little out of the box. Of course, we know that phenotypic screens are not the only thing in the precision medicine in AML. We are seeing the genomic screens as well. However, in the comparison, what we are seeing is that the genomic screens take quite a while, usually weeks to months. And about 90% of the mutations are not targetable. And even those that are targetable might not be available for the patient at that point in time. As I said, phenotypic screens can be done much shorter. However, we have had quite a bit of pushback clinically for historical reasons, because historically these screens had fairly low assay quality. The libraries were very small. So typically the patients got suggested something that they would have received anyway. So there wasn't much of a benefit from adding this as an additional step. And using our screen, we were able to overcome these issues to produce a very high quality assay that can be done in a short amount of time and allow the treatment stratification. But then again, you have to put that to the test clinically. And while we were able to fairly quickly assemble the pilot cohort, it did not go smoothly from the start. We needed to take several stops to readapt, rethink, and make sure that the clinical implementation and the clinical pipeline was actually working. Because the screens, we know how to do them. This isn't the hard part. The hard part is integrating it into the clinical system, making sure that the treating physicians understand it, buy into it, and actually use it for treatment, and to make it as easy for them as possible to actually have access to these screens. We designed a clinical protocol, the DEPICT trial, that allows us to recruit patients that have relapsed or refractory disease and no alternative life-prolonging therapeutic options. So these are really the characteristics. We can recruit pediatric and adult patients on the protocol. I'm going to show the AML here. And for all of these patients, I want to point that out, is that we used standard of care bone marrow biopsies. So they did not undergo any additional procedures or had any additional appointments other than what they were supposed to get clinically in any case. We just asked that they draw a couple of extra tubes for us so that we had sufficient material. Our pilot population was 12 patients, and unsurprisingly, the drug sensitivity profiles are very, very different between patients. There's a lot of heterogeneity in that population. We know that clinically, so that was something that we expected. However, what surprised us is just as how diverse these profiles are. We were expecting to maybe find one or two classes of drugs that show response in most of the tumors, if not all. We have not been able to find that. Our population is much larger now, and we have still not been able to find that. Every patient is its own disease, and every patient has its own drug sensitivity profile. And that really underscores the need, the clinical need for these techniques, because how do we expect them to have good clinical responses if we are giving everybody the same? So really, these precision medicine approaches can tailor a therapy for this specific patient at this specific point in time. And that is very important because these profiles can change between treatment attempts, and we are seeing that now. To show two of the patients in this cohort, what we are seeing is that the DST profiles are patient-specific, and they also reflect the patient characteristics. So what that means is that a patient that has a more benign disease, meaning long disease-free intervals, good responses to previous treatments, generally has more treatment options available on the DST screen and overall higher response rates, while a patient that has failed every treatment attempt is rapidly progressing in the disease has less treatment options available. So we are seeing much higher resistance patterns in these patients. And that seems to be true in all of the different disease sites that we are looking at right now. While clinically the most interesting part is which drugs have the potential to actually kill the tumor cells without killing the patient, of course. What we are also looking at and seeing repeatedly is that some drugs can actually increase tumor growth. The first time we have seen that, it was a bit of an oddball. We were not quite sure if that was relevant or even true. And when we went back to the clinical characteristics of that patient, we could see that the patient that showed proliferation in the wells that were treated with corticosteroids indeed had a very strong progression in response to clinical corticosteroid treatment, which tells us that this is something that needs to be looked at, and it needs to be transmitted into the clinic to prevent a patient from potentially getting a treatment that makes his disease worse. And we don't want that. That being said, while we have been able to find compounds that kill the tumor cells in every patient we have tested so far, these increases in proliferation are fairly rare. We don't see them often, and similar to the killing, they're very patient specific, and there is no class of drugs that will routinely do that. It's really all over the screen, all over the compounds. If it shows up, it shows up. However, like you see it here in the case of the corticosteroids, it tends to be more than one compound of the same class if we are seeing this effect. So this is the outcome of the AML pilot trial. You see in green on the left, the DST guided results, and on the right, the physician guided. So we generated DST profiles for all of the patients, but only the ones on the left were treated based on the DST screen. The ones on the right were purely treated based on clinical criteria and the choice of the treating physician. What was very obvious is that none of the patients on the physician-guided arm had any treatment responses. I want to remind you, these patients are very, very sick. We are basically, as said, as it sounds, scraping the bottom of the barrel here. So they're all very sick. So treatment responses tend to be rare in that population. In the DST-guided population, however, we had 3 patients that showed treatment responses. One had a clinical response. The other 2 had responses either in the bone marrow, a reduction of blasts, or in the peripheral blood, a reduction of blasts. And we saw a slight increase in survival in these patients. I mentioned before, this pilot population has taught us a lot of lessons and made us adapt everything a lot more than we had anticipated. And one of these lessons was that we have to be fast. We have to be as fast as possible. So in patient 3, it took us a week from getting the sample to starting the treatment. Unfortunately, that patient had progressed. And just in these 7 days, and he had a quite significant progression, the doctors decided to still try the DST therapy because there was nothing else in the picture. And the patient showed no response. What really we have to try is to keep the time frame short or exclude patients from clinical implementation that have progressed in that short time frame it takes us. What is reassuring is that we are faster or just as fast as the recruitment on a clinical trial or the other therapeutic options that these patients have. So we were able to achieve that clinical timeline and really give the patient options that he might not have had otherwise. A quick summary just about everything I've said. It is a learning curve, and we need to, in the big clinical trial, really figure out how strict the parameters have to be in order to get the maximum benefit out of these testings. But this small population and the additional patients that we have tested show that DST guided patient stratification is beneficial for these patients. And we need to find a way of making it available for more of these AML patients on a larger scale in the clinic. So yeah, we know it works fairly well in AML, and the larger phase 3 clinical trial is in development and hopefully coming very, very soon to answer some of the questions that are still open. In the meantime, we transitioned the platform to solid tumors. And again, it's a learning curve. I went into it maybe a little naive, thinking it's the same DST screen. It shouldn't be all that hard to transition it. It's a lot harder than I thought. It can be successful, but it requires numerous adaptations. They're not big, but there are many of them. And it really adds an additional layer of complexity to the whole screening approach and to the clinical implementations. The first drawback we have seen is that no liquid biopsies. And the biopsies that the patients are getting, the needle biopsies, they don't yield by far enough tissue what we need. So we need surgical samples for the sample volumes that we need. However, they're not routinely available. Most patients only get one surgery. They might get a debulking surgery later on, but it's not like a bone marrow where you can just go get a sample every 3 months. So sample is really, really limited. And oftentimes they're also needed for other diagnostic tests. So samples are precious, they tend to be small, and things need to be prioritized specific for the patient. It also requires a lot more additional sample processing than the AML samples, which can be purified just using FICO. And different malignancies, even different subtypes of the same malignancies, need to be implemented separately. There's no one-fits-all approach for this. And I learned that the hard way in sarcoma, where you're expecting a piece of tumor and you're basically getting a huge chunk of femur, and you're trying to figure out how to get these cells out of there. Although initially we had tested it on PDX models, that is of course nothing you can experience there. You have to experience this in a patient sample, and you have to deal with that in a patient sample. Other things that we realized with osteosarcoma is that these cells like to put calcium deposits down, which completely messes with the readout of the screen, with the sample preparation, all of the above. So there are a lot of challenges that have to be overcome for every single tumor subtype that you implement. We were able to do it, and I'm showing on the sarcoma side here the osteosarcoma and the Ewing sarcoma. We have also implemented it for esophageal cancer, which is not in clinical use yet. And for ovarian cancer, which is starting to get into the clinic now. We are able, for osteosarcoma and Ewing sarcoma, to reliably generate drug sensitivity profiles for the patients. The screening procedure is quite different from what we're using in the AML. As I said, lots of adaptation. But we are now able to use this clinically in pediatric and adult patients. Similar to what we have seen in AML, the drug sensitivity profiles are patient-specific, and the earlier in its journey the patient is and the better he responded to prior therapies, the more treatment options we have. Ewing sarcoma generally is a harder to treat tumor that shows high levels of resistance and doesn't respond all that well to therapy, and we are seeing that in these screens as well. We are nearly done with the pilot populations for osteosarcoma, which will hopefully be completed in the next 1 or 2 months, so that we can look at outcome and overall benefit of the DST screen for these patients. However, we have especially our poster child patient, which had a phenomenal response to the DST guided therapy where it was implemented. So we are encouraged to continue this in these tumor types as well. What we have seen is that specifically in the pediatric population, there is a lot of interest for approaches like this, because these patients progress fairly rapidly, and toxicity is a major burden for the children. So possibly even with lifelong consequences, because if you're having differences in bone growth or in blood cell development, this can have lifelong consequences for these patients. So having the toxicity output is really valuable for the pediatric doctors here. The platform can be transitioned to solid tumors. There are adaptations. The biggest hurdles that we have seen is that every tumor subtype has to be implemented separately, which takes a couple of months. It is doable, but it takes time. Sample collection and processing are very, very different and have to be implemented and need an extra step of implementation on the clinical level, just to make sure that in the OR, in the pathology, everything is handled in a way that allows us to do the screen downstream. Media and seeding densities, of course, also have to be adapted. But one of the big issues that we have had is sample size. We need a lot of tissue to run the full screen. Oftentimes that is not available. So patient-specific libraries are becoming more and more important. On the other hand, for example, the pediatric populations, a lot of drugs, even though they're FDA approved, are not available in that population. And it doesn't make sense to run the screen using chemotherapy drugs that the patient has no chance of ever getting. So we have to adapt the libraries to our patient population to make sure that the screen can be run optimal and we get the most benefit for every single patient that we are testing. This is a big undertaking. And of course, I haven't done it by myself. We have a DST screening team that deals with the day-to-day DST screening. And we are currently actually expanding that team because we have more samples than we currently can handle. As I mentioned, Dr. Hassan Al-Ali has been the brain behind the DST algorithm and the analysis that we are doing. He's responsible for that, and he's a very important part of our team. But of course, we also have very, very supportive clinical collaborators that make sure that we know about patients, that recruit the patients, that deal with the treatment implementation, and that they're just generally very open to what we're doing and are very supportive of the whole concept. We had funding from multiple different sources, most recently from the Sebastian Strong Foundation and the Coulter Foundation, who are allowing us to expand the screening platform, automate it further than what it is now, and to further develop the analysis and the screen for different malignancies. So yeah, we are very thankful for their support. Thank you.
Great, this is Chris. I'm pinch hitting for Tony, who had to step away for a second. So thank you very much. That was very exciting. We have a few questions. First of all, from our colleague here, is this a CLIA approved test?
No, not yet. We are working on that.
And so Keith Ligon is asking this question. So maybe you could clarify that. Is this within a clinical trial, or how do you provide this information to doctors? Then I'm just a little curious how that works.
So this is all done within a clinical trial. We have the PIC protocol that was made specifically for this purpose, and all patients get enrolled in the clinical trial. And we are receiving consented material from consented patients that our own doctors have consented, of course. If they're coming from the outside, they're still going through our doctors for consent and enrollment. We receive the samples, we are running the screen, and we are giving the information to the PTC, which then transmits it to the treating physician.
Thank you.
Do you have, if I could just submit, do you have an investigational device exemption from the FDA to perform this?
No, for the pilot trials, we don't have. We are working on this right now. Because for the large clinical trials, that is needed.
Can you talk a little bit more about your QC metrics?
So the QC metrics are multi-layered. Of course, when we receive a sample, we are looking at sample quality, sample size, viability, all of that. It needs to have been shipped in the appropriate conditions, otherwise it's not accepted. It has to have a certain level of viability. We're preferring 90% or more. And it has to have a certain sample size. If you're sending us 1 milliliter of blood, that obviously isn't useful for what we are doing. The next quality control layer comes after the purification, again, where we are looking at viability, sample quality, all of these measures that are very important for the success of the screen. After the screen is run, the quality control involves looking at the positive and negative controls. Are they run, are they within parameters on all of the plates of the screen? Because we are using multiple assay plates. And we have duplicate compounds on the screen so that, for example, gemcitabine is, and I'm pulling that out thin air, is on plate 1 and plate 10. And we have to make sure that the sample responds the same way on both plates. If there is too much of a deviation, the screen is a screen failure and is not further analyzed. After the analysis, of course, all of the curves that are generated are QC'd to make sure that the results that are coming from the algorithm look the way they're supposed to look, and we're agreeing with them. And then as a last step, all the quality controls are looked at again to ensure that the entire screen, everything was looked at least twice, and everything is within parameters before it ever gets transmitted anywhere.
What's your percentage of success from initial biopsy to a successful screen?
We are currently at 96%.
And what's your total number of samples so far?
Over all of the disease sites?
Sure, yeah. Mm-hmm.
We are over 50.
Oh, great. Congratulations. That's hard work. I know how hard that is. That's amazing.
It's a learning curve. It's for
great.
sure a learning curve.
Fantastic. What normal cells do you use as a control? You said what are the normal cells?
That depends on the tumor type. We are using matched controls, but we have a database of controls. For example, white blood cells, normal osteoblasts, normal osteoclasts, fibroblasts. We have a whole range, and we have them in pediatric and adults so that we can match to the disease site and also to the age of the patients, which we found to be important. What we are trying to find out right now is if we also have to match for ethnicity, but that analysis hasn't been completed. That is part of the large clinical trial to make sure that that actually works. So, and what we also see is that some doctors, especially if the patient has a very complex disease, request normalization to different normal controls so that they can find the least toxic option for their patient. For example, if you have a neoplastic disease, also testing against the white blood cells is often a good idea in our experience.
What volume of tissue do you need? This is from Ken Dixon. Or number of cells, that we could say as well. One of those two metrics.
That depends on the tumor type, sadly, because our seeding densities change. For AML, we typically ask for 20 milliliters of bone marrow, and that seems to be sufficient. We had patients with such a strong disease load that 10 milliliters was more than what we needed. For our sarcomas, we are asking roughly for 1 centimeter by 1 centimeter pieces. But again, different tumors are needing different tumor sizes. And we also request pieces that look viable in during the surgery, because if half of the thing is necrotic, that first of all doesn't give us enough cells. And we have also seen that it compromises the viability of the remaining cells in the culture.
Okay. I think the questions on tissue specificity and age of the patients were answered. And we're a little beyond time. Does anyone else have any burning questions that they want to ask before we sign off? No? Well, that was really exciting. I really want to thank both speakers for really fantastic presentations and pushing the limits on functional precision medicine, and keep working hard at it. It sounds like we're all making great progress, and can't wait to hear updates from both speakers in a couple years or a year or so from now to see where they are with these platforms.